Introduction. Hi everyone, I'm Chen from the Department of Material Science and Engineering. Today I'm glad to be here to talk about my research about using this iohexol nanoparticle used as a contrast agent for the in vivo X-ray computed tomography imaging, also called CT. So as everyone knows, the cancer is a deadly disease. In the past several decades, a lot of people tried every method to fight with cancer. In this long and tough fight, so people gradually realize the importance of the early diagnosis. The earlier the cancer can be detected, the better chance it will be killed. So among a variety of the diagnostic tools, CT is one of the most powerful tools for the diagnosis. Because of its low cost, it has been widely used in the clinical today. So here is the scheme to illustrate the basic principles of the CT scan. So here you can see the CT scan made by the rotating X-ray beam around the patients. So by collecting imaging either horizontally or vertically, the CT scan can generate a series of slice images and stick them together. So you can imagine the generated CT images just like a loaf of bread. So the bread, after the imaging, the doctor can either take one slice of the imaging to look at the 2D dimensional imaging, or look at the whole loaf of bread. That means they can look at the construct the three-dimensional imaging. In this case, the CT scan can provide the detailed information about the cancer patients in terms of the size of the tumor, also the location of the tumors. So here is, I just take a CT scan from the website. So here it shows the CT scan of a brain of the glioma. So by looking at this figure, we can see that the doctor can, based on this imaging, to locate the location of the tumors and also to determine what the size of the glioma in the cancer patients. And based on this basic information, the doctor can give the related treatment. Nowadays, the current clinical use CT contrast agents are largely based on these iodinated small molecules. One of the examples is called the iohexol. Because of the iodine has a very high Xray absorption, so it can provide the very good CT contrast in vivo. However, because the low molecular weight of the small molecules after injecting into the body, they tend to be faster eliminated from the body, resulting in a very narrow imaging window. In this case, it will bring some of the undesired effects for doctors. For example, because the imaging window is so narrow, sometimes the doctor may miss some of the very small but very critical the metastatic lesions. As we all know, to determine like the development of the cancer, sometimes we need to get the information, especially for these metastatic lesions. Also, because the faster clearance from the body, especially from the kidney, it always brings such the severe toxicities, such like the severe renal toxicities. To overcome these problems, in the past several decades, a lot of researchers put a lot of efforts to try to develop new nanoparticle-based CT contrast agents. So one example is here. So they can use this liposome to encapsulate these small iodinated small molecules into the liposomes. However, for this typical system, always because the loading is so low, it cannot provide the sufficient contrast for the doctors. And also the burst release of small molecules after injection always be observed. So in these typical systems. And also after that, a lot of researchers also try to develop new inorganic-based nanoparticles. For examples, I listed here. So people use these gold nanoparticles or these polymer-coated bismuth sulfate nanoparticles, or either these upconventional nanoparticles. But as we all know, like the long-term toxicities about these inorganic particles always be a huge concern, especially for the further clinical translations. So by comparing the about two systems, either these small iodinated small molecules or like these inorganic nanoparticles, we can help asking ourselves, can we just combine these strengths of the both systems together to make a better systems, either with the high X-ray absorption or have the establish the good the biocompatibility, or we can take advantage, or at the same time we can take advantage of the properties of nanoparticles, such as like elongate circulation and protracted the tissue retentions. So it is possible to just derive from these iodinated small molecules as components to construct a nanoparticle system to combine these both together. So by noticing that these iohexol small molecules have a variety of the functional groups, for example, it has 6 hydroxy groups, it will be possible for us to further manipulate. So in this case, we come up with very straightforward and simple strategies to use this hexamethylene diisocyanate as a cross-linker, just cross-link these small molecules to make these cross-link polymers. And then by nanoprecipitation with this PEG-PLA, widely used the diblock polymers, we can formulate these pegylated nanoparticles. So with these systems, we hypothesize that the new system could outperform the clinical use contrast agent iohexol by substantially either elongates the circulation and also at the same time to improve the tissue retention. The whole strategy is really simple but very efficient. So first, we demonstrate that using this strategy, we can very well control the molecular weight of the cross-linked polymers. For example, by changing the ratios of the iohexol or isocyanate by using the different ratios, we can see that the GPC traces, we can get a series of the polymers with different molecular weights. So we notice that the polymer made as a ratio 1:3 can get with the largest molecular weight, like as high as 68 kilodalton. So as we all know, like the larger molecular weight always bring like the renal clearance rate. In this case, the P3 polymer will be chosen as the candidate for the further exploration. And also we demonstrate by using this nanoprecipitation method, so we can formulate these nanoparticles with very good physical and chemical properties. For example, here we use the dynamic light scattering and also the SEM to show that the formulated nanoparticles have the size around 150 nanometers and with a very narrow size distributions. And also the nanoparticles formulated show the remarkable stabilities against a variety of the physiological buffers. For example, we can see that either in the PBS or in the human serum, nanoparticles still keep its size without any aggregation formed in these both the physiological buffers. So most importantly, if we want to translate these nanoparticles into the clinical translations, we need to demonstrate it could be retain its properties during the either storage or the transportation. So here we demonstrate that after the lyophilization, the solid form nanoparticles can be redispersed into the water, still keep the nanometer size range without any large aggregates formed. In this case, it shows a great promise for the later clinical translation. So as a candidate for the contrast agent, the most important thing is his X-ray absorption, whether they can provide the efficient contrast. So here, first we did this ex vivo study. So we collect the CT scan about a series of the nanoparticles with different concentrations, and then convert them into the HU units for the quantifying. So this figure shows that the CT attenuation, these HU units show the linear relationship with the different concentration of nanoparticles. In this case, we can draw the conclusion. This nanoparticle is possible for the quantitative CT analysis in vivo. CT analysis. So how about the in vivo performance? Then we go ahead to do this in vivo study. So we inject these nanoparticles into these New Zealand white rabbits through this transarterial injection about the mixture of the nanoparticle solution and also the proxy seed oil together. So compared to this CT scan taken before the injection, we can see that after the injection, the large enhanced has been observed in these small liver modules. And also because a large amount of nanoparticles still retain in this portal vein, so we still can see enhanced contrast in these vasculatures. So in this case, it will further indicate that the nanoparticles may potentially can be used to detect some of the vascularized, the hepatic, the metastatic disease. So with these good properties and the in vivo performance, we are so excited to move forward. So here we established a new tumor model is MCF7 human breast cancer bearing arsenic mice. So here we carry out a time course study by injecting the nanoparticles through the intratumor injection and then collect the imaging at the different selected time points to evaluate the enhanced contrast in the tumor regions. So here we can see that after 5 minutes injection, the mice treated with iohexol actually show the pretty good contrast in the tumor regions. However, the signal decreased so fast and become fairly weak at 1 hour and even non-detectable at 4 hours in the tumor regions. However, for the mice treated with polyiohexol nanoparticles, we can see that after injections, the contrast obtained shows the comparable contrast compared to the iohexol, but the signal retained very well. So after 1 hour and even after 4 hours, we still can see the enhanced contrast in the tumor regions. In this case, it's kind of demonstrate our hypothesis. We can use this to outperform the small molecules, show the improved the tissue retentions. And then this quantitative results further confirmed the observation. So here, this we quantifying the tumor density means we get the contrast in the tumor region and subtract the original contrast. Then we can do this figure. So by comparing the small molecules, we can see that at 1 hour, the nanoparticles shows like the 3-fold compared to the small molecule, and even as high as the 36-fold compared to this one. So in this case, the result is really striking. And then when we look at the mice treated with both contrast agents, we can see that at 5 minutes after injection, the bladder already been lit up by the small molecules. However, for this nanoparticle, the signal is fairly weak. In this case, it indicates that the slow clearance rate of the nanoparticle compared to the small molecules. So after that, so we keep asking ourselves, can we use this for further studies? So what kind of studies we can perform? So next, we want to move forward to do the dynamics CT. So as a preliminary concept proof, so we just use a fluoroscopic imaging to capture the CT scan during the injection as long as 60 minutes. So here are the two movies to show the mice treated with two contrast agents. So the left side is mice treated with nanoparticles through the jugular vein, systemically administered nanoparticles. We can see that after the 5 minutes injection, the bladder already shows a very high enhancement, and the signal becomes stronger and stronger. However, the signal retained in this heart and the liver and this kidney become gradually decreased. However, for these mice treated with nanoparticles, we can see that the bladder still keeps non-detectable for the whole imaging process. So this big difference between the contrast in the bladder of the small molecules and the nanoparticles is further demonstrated that even with the systemically injection, we can show that the elongates the circulation and the slow clearance of this contrast agent. So here, I just take the snapshot at two different time points to compare. So one is at the 5 minutes injection, and another is at 1 hour injection. So here, by using the image J, I can quantify the enhanced density in the variety of organs. So here, I just showed the enhancement in the liver and in the heart and the liver. So we can see that the signal in the heart and the liver didn't show the much decrease for the treatment with the treatment of the nanoparticle contrast agent. However, for the treatment of the small molecules, we can see the large decrease compared to the original injections. In this case, we further demonstrate our hypothesis, so the elongated circulation and also protracted tissue retention. So, and then we performed so next we want to confirm like how the nanoparticle is circulate as we expected to show the elongated circulation compared to the small molecules. In this case, we use a copper-64 to label both the nanoparticle and the small molecules and inject them into the mice through the IV injection. And then we take the blood at the selective time points to measure the radioactivity in the blood, and then thus we can get the concentration of the either nanoparticles or the small molecules in the blood. So here we can see that at the first 30 minutes, both of them show the large drops. However, the concentration in the blood of the nanoparticles still much higher than the small molecules, almost twofold higher than them. And then by applying this equation published in the Nature Nanotech paper, we can get the in vivo half-life of both systems. Like here, I indicate the small molecules is around 4 hours, and the polymeric nanoparticles is as long as 16 hours, almost fourfold than the small molecules. In this case, also, we use another study to demonstrate our hypothesis, elongated circulation of the contrast agent in the blood. So as a contrast agent, we do not want them to perform as a drug to stay in the body so forever and do not clear it out, because usually the patients go to the hospital to take the imaging scan to get the disease diagnosis, then they need to go back home. So in this case, we performed the clearance study to see whether the nanoparticle the developed nanoparticle system can be cleared from the body and do not leave any toxicities to the body. So by doing that, we use the copper-64 to label both systems. So here I just show the final structure to show how to label them. And then by taking advantage of the PET-CT, we can show that at the different time points, the accumulation of the nanoparticles and the small molecules in the mice. So here is the tumors. So we can see that by comparing to these small molecules, nanoparticles show the very good accumulation in the tumor regions, presumably because of the EPR effects. And also, so after 24, and also based on these imagings, we can get the quantifying the accumulation in the different organs. So here I list as different organs such as the heart, spleen, liver, lung, bladder, and tumor. So we can see that for almost all of the organs, the nanoparticles show the higher accumulation than the small molecules, especially except for the bladder. They show the comparable accumulations. So in this case, we can see that by using these larger size nanoparticles, so we can achieve the good retention in a variety of the organs. And after 24 hours, we take out the different organs, use the gamma counter to further quantify that. So here we can see that we see the large accumulation of the nanoparticles in the urine and the feces. So it demonstrates the nanoparticles can be bioeliminated from the body through the urine and the feces. And also because the iodine always relates to the hypothym. So here we show that the low accumulation in the thyroid to further demonstrate the deiodination is not the major problem about this newly developed system, to further confirm the safety profile of this one. So last but not least, so as a candidate for the further clinical translation, the biocompatibility always like the very important parameter to measure. So here we just use the histological evaluation to evaluate the safety using this by injecting this nanoparticle into the mice. And after 24 hours, we take out the different organs, do the HE staining, and to compare with the mice treated with the PBS. So with the pathologist's help, we can see that no any major organ damage have been observed in this polymeric nanoparticle treated tissues compared to the PBS treated tissues. In this case, so we can show that the low in vivo acute toxicities about these systems. So in conclusion, so by using a very simple and straightforward strategy, we can formulate this in vivo applicable CT contrast nanoprobe through cross-linking the small molecules, and following by the co-nanopreparation with the diblock polymers. And these nanoparticles shows a negligible in vivo toxicity, remarkable stability, and substantially improved circulation and tissue retentions. Especially, it could be possible to elongate the time window for the more accurate diagnosis of the patients. And also, because the whole strategy is so simple, so it can be easily scaled up and formulated into the solid form with well-preserved properties, without any the aggregation have been observed in the whole process. In this case, it could be used as CT contrast agents in the clinic to improve the diagnostic accuracy and patient compliance. So at last, I would thank my advisor, Jian Junchen, and also my co-advisor, Tim Fan, for their tremendous help. And also, I would like to thank the collaborators in the campus or out of the campus. So at last, I want to especially thank Laura for her help in the past. So thank you very much. I would like to take any questions.
So there was a difference in the absorption spectrum before and after lyophilization.
Mm-hmm.
Do you know why it occurs?
Yeah, because you see we use the PEG-PLA to help to stabilize the nanoparticles. However, by using these diblock polymers, based on our experience, we always can see the size increase. But without this pegylation, usually like other systems after the freeze dry, you cannot disperse them without any aggregation. So here, even we can say like the 20 to 30 nanometers size range, but still keeps the nanometer size. So in this case, we can inject it into the body without bringing any like the instant death to mice. Mm-hmm.
You mentioned that the plasma half-life was around 16 hours for the nanoparticles.
Yes.
How could you regulate that time? I guess could you make it shorter or longer? And what are the ways that you?
Oh yeah, because here, like I said, so we use the PEG-PLA to stabilize a nanoparticle. PEG is a hydrophilic polymer used to elongate the circulation. So I guess by changing the different ratio of the PEG-PLA compared to the cross-linked polymers, if you make the dense PEG, you can elongate the circulation. If you make the loose PEG, probably you can reduce the circulation. Because also as a contrast agent, we want to in vivo like the circulation life in the certain range. Because if it's too short, just cleared from the body without providing the sufficient contrast. But if it's too long, like more than 1 day, it's not desirable because you need to be cleared and then send patients home. So in this case, I remember some of the researchers suggest that I think the 1 day is a good time range for this contrast agent platform. Mm-hmm.